In this video, we are going to tell you about lysosome or suicide bags of the cell. We are going to focus on what is meant by a lysosome, its structure, who have these organelles, its synthesis and polymorphism, lysosomal enzymes and lysosome functions. What is meant by lysosome? In Greek language, lysis means digestion or breakdown and soma means body. This means we can call it as the cleaner or the janitor of the cell. Lysosomes can be found in the cytosol of the cells, floating freely within the cells outside the nucleus. Their shape varies depending on time and cell type called polymorphism, but mostly amoeboid or white blood cell, or WBC, shaped to spherical-shaped vesicles or sacs. Their size also varies ranging between 0.1 to 0.5 micrometer. This is a simple organelle with a single membrane. Its outer membrane is surrounded by an acidic interior fluid, which is made up of hydrolytic enzymes or hydrolyses and dense material. Its pH is in acidic range, which is between 4.5 to 7.2 with an average of 5.0. This low pH helps breaking down macromolecules and larger cell debris easily and provide the enzymes a nice active environment for reactions. Lysosome has two main parts, a lysosomal, or limiting, membrane and an inner dense mass or matrix. The membrane is a single membrane of lipoproteins with acidic and highly glycosylated integral proteins and protein transporters, such as H plus ATPase. The inner dense mass or matrix is granular, dense, and heterogeneous ground substances. Some lysosomes have a very dense outer zone and a less dense inner zone. Some others have cavities or vacuoles within the granular material. Now, let's make a definition for lysosome. Lysosomes are small sac-like membrane-bound cytoplasmic cell organelles that contain hydrolytic enzymes for cellular digestion. Let's go back to the history of the lysosome to see how it was found. During electron microscopic studies in the early 1950s, rounded dense bodies were observed in rat liver cells. These bodies were initially described as paranuclear dense bodies. Later in 1955, Belgian cytologist C. de Duve renamed these organelles as lysosomes. Lysosomes can be found in all nucleus-bearing, or eukaryotic, cells. Mainly in most animal and few plant cells, such as onion seeds, barley seeds, corn seedlings, and also in some fungi, such as yeast, neurospora, and so on. These are absent in prokaryotes such as bacteria and mature mammalian erythrocytes. Leukocytes, especially granulocytes, are rich in lysosomes. Few lysosomes occur in muscle cells or acinar cells of the pancreas. Let's get to know about polymorphism of lysosomes now. Due to their extremely dynamic nature and shape in one cell or different cell over a different time, lysosomes are polymorphic, means lots of morphologies. Polymorphism of lysosomes depends on the type of substance that has been absorbed into the cell by phagocytosis. Generally, lysosomes can be traced in four forms, including primary lysosomes, secondary lysosomes, autophagosomes, and residual bodies. This slide shows you a summary of all these four types of stages in lysosome bodies. Primary lysosomes are also called storage granules or protolysosomes or virgin lysosomes. First, lysosomes are produced in Golgi apparatus. Newly formed ones are primary lysosomes, which contains enzymes in inactive forms. Secretes enzymes out of the cell or stores inside itself. This is not involved in digestion. Secondary lysosomes are also called heterophagosomes or digestive vacuoles or heterophagic vacuoles or phagolysosomes. These are formed by the fusion of primary lysosomes with endosomes. You can find them as phagosomes and pinosomes or cytoplasmic vacuoles containing extracellular substances. These are involved in cellular digestion. Autophagic vacuoles are also called autophagosomes or cytolysosomes or apolysosomes. These are formed when the cell feeds on its intracellular organelles such as the mitochondria, endoplasmic reticulum, or Golgi apparatus by the process of autophagy, or autodigestion. Lysosomes are concentrated around the intracellular organelles and digest them. These are actually a type of secondary lysosomes. Fourth type is residual bodies, which are also called as telolysosomes or dense bodies. Lysosomal with undigested materials are called residual bodies. After complete digestion in heterophagosomes and autophagosomes, some substances remain indigestible grains or amorphous masses or ferritin-like or myelin figures. Now let's get to know about lysosomal enzymes. These are synthesized in the endoplasmic reticulum and passed on to the Golgi apparatus where the lysosomes are produced. The lysosomes use the acid hydrolysis from the endoplasmic reticulum to digest complex proteins and organelles that are no longer needed. Around 40 types of hydrolytic enzymes have found so far, such as proteases, nucleases, lipases, glycositosis, and so on. All lysosomal enzymes are acid hydrolyses that function more efficiently under slightly acidic medium. These are optimally active at pH 5.0. Here we have listed some types of lysosomal enzymes, including nucleases, proteases and peptidases, phosphatases, enzymes acting on oligosaccharide chains of glycoproteins and glycolipids, enzymes acting on glycosaminoglycans, and enzymes acting on lipids. Lysosome can perform important functions in a cell and in an organism, such as intracellular digestion, extracellular digestion, role in metamorphosis, help in fertilization, removing dead cells, lysosomal storage diseases, hormone secretion, and other lysosomal functions. During intracellular digestion, lysosome can digest or destroy its own cell organelles, which leads to the death of the cell. This is called autolysis, and that is why lysosomes are named as suicide bag of the cell. Depending on how this digestion happens, we can find two types of digestion: heterophagy and autophagy. In heterophagy, extracellular materials and heterophagosomes are digested by the lysosomal enzymes. This is important during defense mechanisms, food digestion of protozoans, etc. Mainly two types: phagocytosis, pinocytosis. In autophagy, various intracellular organelles or materials in the autophagosomes are digested by the lysosomal enzymes. Autophagy mainly occurs during critical cell conditions such as pathological conditions, starvation, stress, also known as autolysis. Autolysis helps in the regression of the uterus following delivery, the regression of mammary glands following weaning. Both heterophagy and autophagy may occur simultaneously within the same lysosome. Such a lysosome is called the ambylysosome. In the second function of lysosome, or extracellular digestion, some cells release lysosomal enzymes to the outside through exocytosis for the digestion of extracellular materials. During the remodeling of bones, the osteoclasts of bone undergo extracellular digestion of the bone matrix. Also, saprophytic fungi obtain their nutrition through extracellular digestion. Lysosome also plays a role in metamorphosis of frogs and other insects. The disintegration of the tail and gills of tadpole larvae of frogs is due to the activity of the lysosomal protease enzymes called cathepsins. Lysosomal enzymes helps in fertilization too. This whole reaction is called acrosome reaction. During fertilization, the acrosome, a giant lysosome of the sperm head, secretes lysosomal enzymes called lysins. Lysins dissolve the limiting membrane around the egg. A channel is formed, through which the sperm nucleus penetrates into the egg. Lysosomes also play a role during dead cell removal in the outer layer of skin and mucous membrane linings of the body. They also helps to remove bacteria in white blood cells. Lysosomal hydrolyses are involved in secretory actives of some cells. They can modify products synthesized from the cells before secretion process initiates and also regulate the secretion process. For example, during thyroxine formation, lysozyme enzymes are involved in the hydrolysis of thyroglobulin and helps to produce thyroxine or tetraiodophyronine, or T4, and triiodophyronine, or T3, by thyroid gland. Then they are secreted into blood capillaries. Lysosomal storage diseases, or LSD, are genetic diseases caused by the accumulation of undigested materials, polysaccharides, lipids, due to deficiencies in lysosomal enzymes. So far, around 50 types of lysosomal storage diseases are known in humans, such as Pompe disease, Tay-Sachs disease, Gaucher's disease, Hunter's syndrome, and so on. Lysosomes plays a role in cell division as well. Lysosomes stimulate mitotic cell division by overcoming agents that cause repression of mitosis. Further, they play a role during the formation of bone cells from cartilage and thus involves in osteogenesis. There are numerous functions played by lysosomes, and here we're showing some more examples. This is another summary of the functions of lysosomes. We hope you got a good knowledge on lysosomes from this video. Thank you so much for watching. Subscribe us to watch more videos like this, and don't forget to let us know your opinions.